Okay, so good afternoon, everybody. First, I would like to thank to all of you for participating in this seminar, and particularly, I would like to thank to Giovanna Barba Spaeth for accepting our invitation to giving a talk in this seminar series. Today is the first seminars from this for this cycle of seminar. Dr. Barba Spaeth is senior researcher at Pasteur Institute in Paris and the group leader of the FlavImmunity team. She is also coordinating the European project Yellow for Flavi. Her research line focuses on the study of the envelope glycoprotein and the mechanism of viral entry into the host cell. And she's going to share with us a very interesting presentation entitled Flavivirus Immunological Crosstalk, the impact of vaccination. So thank you, Giovanna, for accepting participating in this seminar second, and the floor is yours. So when you want to start, go ahead.
Thank you, Jörg, for the kind invitation first, and then I'm share the screen. See. Okay, I think it's okay, and I put laser point. Okay, well, thank you, everyone. Thank you for joining this webinar. I'm Giovanna Barba Spaeth from the Institute Pasteur, and today I'm going to talk about Flavivirus Immunological Crosstalk and the impact on vaccination. But before doing that, I thought I'd give you a little bit of a snapshot of my scientific background. I'm Italian. I did my undergraduate studies in the University of Palermo in Italy, and I moved to Germany for my PhD. It was a work on the human complement C4. And then for my first postdoc, I moved to Paris. I became interested in virology. I started to work on hepatitis C virus. And then I moved to study hepatitis C virus for a second postdoc to St. Louis, joining the lab of Charlie Rice, and moved with him to the Rockefeller University in New York 3 years later. In New York, Rockefeller, I became interested in flaviviruses in general, but in particular, I worked with yellow fever and the yellow fever 17D vaccine. And I brought the project with me when I came back to Europe to join the Institute Pasteur. I joined the lab at Institute Pasteur. I joined the lab of Felix Rey because I want to complement my training in molecular virology with some notion of structural virology. And in my group, FlavImmunity, we study flavivirus, in particular yellow fever virus, and we are interested in understanding the molecular determinants that drive the conformational changes of the envelope protein of these viruses, and also how the structure impact the epitope exposure and antigen presentation in these viruses. So flavivirus is a genus of pathogenic viruses. They are arbovirus transmitted either by tick, like TB, tick-borne encephalitis virus, or Powassan, or by mosquito, like dengue, Zika, or yellow fever. Yellow fever is the most pathogenic virus of the group. It's endemic in South America and Africa. There is an outgoing outbreak in South America in 2024 going over 2025, which is characterized by high fatality. It's involving endemic area, but it's moving also to non-endemic area. The fatality is reaching 50%, and this has pushed the Pan-American Health Organization to issue an epidemic alert in February 2025. The spread of the virus can be controlled by the availability of a highly efficient vaccine, which is a live-attenuated virus, the yellow fever 17D. These viruses are present, the arbovirus are present in tropical area, but they are moving more and more to a more temperate area. And concerning Europe, we often are confronted with outbreaks of other flavivirus like West Nile or dengue. West Nile in particular arrived in Europe 20 years ago and is now endemic in region of Southern Europe. Every year, a region like a country like Italy and Greece are confronted with outbreak of several cases of West Nile and also associated fatalities. For dengue, last year was a particular year for dengue because there was a worldwide outbreak with 14 million cases worldwide, 10,000 deaths. Of these 14 million cases, 10 million were detected in Brazil, but then via also tourism and traveling, they were imported in Europe, and there were outbreak in France, and there were outbreak in Italy. In Italy, in particular, because the mosquito is present, there have been several cases and outbreak of autochthonous cases. Of the old cases of dengue detected in the fall of 2024, 1/3 came from autochthonous cases, and there was an outbreak in a particular region, the Marche, with 145 cases. This, of course, raises concern of the fact that once the virus arrives in the country, because of the geographical condition and the climate condition, it can spread autochthonously in the country. Contrary to yellow fever, there is no vaccine for West Nile, and the vaccines that are available for dengue have limited success. This depends in part also for the problematic of dengue, because in reality, for dengue, we are dealing with 4 different viruses for different serotypes, and not one single virus. So an efficient vaccine against dengue needs to protect at the same time against all 4 serotypes. And structurally similar is also the Zika virus. So what happens is when you have an infection with one serotype, you generate serotype-specific antibody that can neutralize the same serotype, but will not neutralize the other serotype, but will be able to bind this serotype. And because they are non-neutralizing, the serotype-specific cross-reactive antibody may mediate the antibody-dependent enhancement, where the virus antibody complex can enter the cells via the Fc receptor and increase the infection in the body. And this ADE is the cause of the exacerbation of disease in secondary infection. During the infection, we can also develop broad cross-neutralizing antibody, and we have characterized some of them now almost 10 years ago. And this antibody target a specific epitope that is linked to the E dimer at the surface of the particle. So, as I say, the vaccine needs to be able to generate immunity against all the 4 serotypes, which was not the case for the vaccine that are available. The first vaccine license was the Sanofi, then Sanofi vaccine, but it was not able to have to give a good immunity and protection against dengue too. And it has been now discontinued. The only vaccine at the moment available is the Takeda vaccine, but also for this vaccine, there is poor protection against dengue 3, which was the virus mainly circulating during the outbreak last year. And so to avoid the ADE, in reality, these vaccines are suggested in subjects that have already got the infection, so they will function as a boost and not really as a vaccine. And the challenge is due to the structural similarity of these viruses. Flavivirus are positive strand envelope viruses. They have an mRNA genome that is translated in a polyprotein, cleaved in 3 structural protein and 7 non-structural protein. The viral particle is formed by the structural protein, and the envelope protein is the protein that is at the surface of the virus, is the one that interacts with the immune system of the host. The envelope protein is organized in dimers. It's 90 dimer at the surface of the virus, organized in a herringbone pattern. And here I'm showing you the structural domain of the monomer of the envelope protein. By convention, the domain 1 is in red, the domain 2 is in yellow, and the domain 3 is in blue. And at the tip of the domain 2, there is a stretch of highly hydrophobic amino acid that constitute the fusion loop, which is important for the viral fusion. So during the cycle, the virus enters the cell via receptor-mediated endocytosis, and then in the endosome, the low pH of the endosome induces a conformational change of the envelope protein. The dimer disassemble and is organizing itself in a trimer. At the tip of the trimer, there is the fusion loop that now is exposed. It can interact with the membrane of the endosome and can mediate viral fusion. The RNA is released, translated, replicated. The assembly replication complex from the first budding particles. The particle buds in the ER, and they bud as an immature particle where the E protein is organized in trimer, and the fusion looping is protected by the other second envelope protein of flavivirus, the premembrane protein, with the portion PR that is here highlighted in green. The immature particle then continue in the membrane, intracellular membrane. When it reaches the trans-Golgi, the low pH of the Golgi induces another change of conformation. The trimer reorganizes itself in a dimer with the PR still bound, and this reorganization exposes a cleavage site between PR and the M protein. There is a furin cleavage site. So the furin of the in the trans-Golgi cleaves PR, and then we have the release of a mature particle with E dimer and without PR. So on this E dimer, PR is not bound anymore, it's not protecting the fusion loop, but the fusion loop is not exposed because it is concealed in the structure of the dimer. The fusion loop cannot be exposed, otherwise the virus will stick to all the membrane and there will be premature fusion, and here the endosome is the only place where where the virus wants to expose the fusion loop. So in the dimer, the fusion loop that is at the tip of one protomer is concealed and protected by the domain 1 and domain 3 of the other protomer. And in particular, there is a region of the domain 1 that is called the 150 loop that leans on the fusion loop like a cap, protecting the fusion loop in the dimer. So when we have the E dimer, there are particular epitopes that are exposed, that are the one that we characterize for dengue and Zika, and that we call E dimer epitope, or EDE, that are targeted by highly potent neutralizing antibody. However, because of necessity during the virus cycle of the virus change conformation, this protein, this envelope protein, is highly dynamic and is in highly metastable state. So this dimer at the surface of the virus is not stable, and it can occasionally open up, destroying the EDE epitope and exposing different epitope, like the fusion loop epitope. And so we can have in circulation viral particle that expose different type of epitopes on its surface. And not only that, another source of heterogeneity is due to the fact that this process of maturation that I have described is not really efficient, and there are secretion of particles that are partially mature and partially immature, and exposing again on the same viral particle different epitope. So the E-dimer dynamics determine antigen exposure. And these are consequences because the type of antibody that are generated by this epitope have different functionality. In the mature particle, as I mentioned before, we have the E-dimer epitope. This is targeted by EDE antibody. So this EDE can bind the entire particle, and this antibody results to be neutralizing. However, the virus is heterogeneous. The virus breathing is how we call this open up of the dimer, occasionally expose the fusion loop epitope. And fusion loop antibody will bind this fusion loop epitope on the part of the particle that expose the fusion loop epitope. Similarly, for the partially immature particle, there are antibodies that recognize the premembrane protein that will bind the viral particle only on this epitope. This results in viral particles that are partially covered by antibody and that can be taken up by cells via the Fc gamma receptor. But once these particles are in the cell, this part of the virus is still able to fuse, so this complex will be infectious. And this is the antibody-dependent enhancement. And so, while ADE antibody are neutralizing, fusion loop antibody are mostly enhancing and cause antibody-dependent enhancement, or ADE. The fusion loop, in particular, the sequence of the fusion loop is highly conserved, so these antibodies are cross-reactive. And ADE is not only occurring in the context of dengue or Zika, but this type of antibody will bind all the flavivirus. And so the immune response that you get it after your first encounter with the flavivirus, and this is basically conditioning the response, the other response that you can that the body can generate when encounter other flavivirus. So we wanted to study the impact of pre-existing flavivirus immunity on antibody response generated by the yellow fever 17D, which is a flavivirus, is a live attenuated vaccine. And we did this in collaboration with Simon Rotenfusser for LMU Munich. And this work from PhD student Antonio Santos Peral. Simon has a cohort of a subject of 250 subjects that have been vaccinated with 17D, and we have followed them during the first month of vaccination. And we have also a small cohort via a collaboration with Kilian Schoder from Erlangen University. That is a small cohort of 20 vaccinee, but we follow them until 1 year post-vaccination. And the characteristic of this cohort, because it's from Germany, TB is endemic there, many of these subjects had received previously the TB vaccination. And in particular, the cohort have 2/3 of the subjects that have received the TB vaccine, and 1/3 was naive. Also, the other cohort, more or less this type of ratio. So when we start to look at the antibody response generated after vaccination, and we look first at the IgM titer directly against 17D, and by convention for the old talk here, the TB vaccinated are represented in pink, and the TB unvaccinated, the naive, are represented in light blue. So the IgM titer in the two cohorts was increasing at 2 weeks post-vaccination with an antibody response directed against 17D. The TB experience had a slower response, but it was still effective. They were still neutralizing. 17D. When we look at the IgG titer, the increase at 2 weeks was lower, especially in the naive. The response anti-IgG, anti-yellow fever in the TB experienced subjects was much more important. Not only that, it was also present at high level at baseline. So even before this subject have seen the 17D vaccine. And this discrepancy in the IgG titer was persisting also 1 year after vaccination, with the TB experience have a high amount of IgG that can bind yellow fever 17. So we analyzed the two fraction IgM and IgG for their functionality, and so we could find that the IgM fraction is the fraction that have the neutralizing activity. If we use IgG depleted serum, so remove the IgG from the serum, we look only at the IgM, we have neutralization of the virus equally for the two cohorts. The depleted serum is in gray, and you see the curve overlap the complete serum. On the other side, if we deplete IgM and we look only at the IgG, we reduce drastically in both cohorts the neutralization. So we can conclude that the activity, the neutralizing activity of 1 month after vaccination is still on the IgM fraction. And this is actually maintained even 1 year post-vaccination, where the IgM fraction is still the fraction that's mainly the neutralizing activity. What about the IgG fraction then? The IgG fraction was the fraction that showed ADE activity. We tested this in vitro, and so the sera of the TB experienced vaccinee show highly IgE, antibody-dependent enhancement against yellow fever. But not only against yellow fever, when we tested in vitro, we could see antibody-dependent enhancement also for infection of dengue and infection of Zika. So the sera of this subject that are here again in pink had mediated ADE for dengue and Zika, where the sera of the naive people that receive only the yellow fever vaccine were not mediating ADE activity. To check, to analyze which one were the epitope target by this ADE activity, we then design a toolbox of antigen based on the envelope protein, where we could separately express different epitopes. And we use this toolbox to dissect the epitope landscape after yellow fever vaccination. Before using on the sera, we verify that our antigen recognize the antibody that we want to recognize, So, for example, we tested with purified and known antibody. And so we did this by checking a shift on size exclusion chromatography. And I show briefly here how we do the test. We have the antigen, for example, the soluble monomer of E that we run in size exclusion. This protein is 50 kilodalton and elute and a certain volume. Then we have an antibody like the 4G2, which is an antibody recognized the fusion loop epitope. This is 150 kilodalton and elute at this different volume because it's a higher molecular weight elute earlier. We mix the two, and we mix the two in solution with an excess of antibody, and we run in a size exclusion chromatography, and then we have two peaks. One that correspond to the excess of antibody, and the other that correspond to the complex protein plus antibody. So these are 200 kilodalton, and then there is a shift. And if we overlap the three components, we see that the peak corresponding to the protein in the complex that is here in red is shifted and is now eluting at a lower elution volume because it's a higher molecular weight. So the different antigen that we designed was one is the soluble monomer, but the protein at the surface of the virus is a dimer. Only that the soluble protein in solution is an equilibrium monomer dimer. So to obtain the dimer, we force the dimer formation in solution by building a disulfide bond. Here, generating a binding, generating a dimer, binding the 2-domain 2. This dimer will then present a E-dimer epitope. But for the construction and the dynamics of the protein in solution, this dimer is not locked, it's open up, it can breathe like, of course, on the virus, so it can, by breathing, destroy the ED and exposing only the fusion loop epitope. Then we change one of the amino acid of the fusion loop epitope. We mutated to kill this fusion loop epitope in the breathing dimer. And we also construct another dimer that we call the locked dimer, where we change the position of the disulfide bond in correspondence where the EDE epitope is. And this dimer cannot breathe, cannot open, cannot expose the fusion loop epitope. And we test them in vitro with 3 different antibodies. One is pan-Flavi4G2, recognize the fusion loop. Another one recognize is the 5A that recognize domain 2, but also the fusion loop. And then we have a third antibody that does not bind the fusion loop, but still bind domain 2. And we combine the 3 antibody with a different protein, and we look for the protein peak shift, which means the antibody binds the protein. And we could see, we could see that all the complex shift with the antibody with a different construct, except the one that do not express the fusion loop. So the 2 fusion loop antibody are not able to shift the construct that do not express the fusion loop, but they are still, the non-fusion loop antibody can still shift this construct. And this is summarized here in this table. So we have a different antigen that express different epitope, and then will be used in depletion experiment in the serum to deplete sequentially different type of antibodies. And this is what I'm showing you here, where we at this point focus on the IgG fraction. We took the first the complete serum, and in this column is the naive, and in this column are the vaccine TB pre-exposed to TB, subject pre-exposed to TB. The complete serum shows ADE, as I showed you before. And in this serum, we have all this type of antibody, fusion loop, dimer antibody, domain 3, virus specific. Now, if we use a construct that has only domain 1 and domain 2, we deplete from the serum the fusion loop antibody. We keep still this antibody, but the serum of TB vaccinated has lost the ADE activity. The same if we use the breathing dimer, where we basically expose EDE epitope, fusion loop epitope. So we remove all the type of antibody present in the complete serum, with the exception of some quaternary virus specific epitope. Again, here we do not have ADE activity. However, if we use the breathing dimer, where we knock down the fusion loop, and so we are not able to fish out at least all the fusion loop epitope, we still leave some fusion loop antibody, then we rescue the ADE activity. So this, when we have presence of fusion loop antibody, we have ADE activity. So pre-existing immunity to flavivirus skew vaccination immune response towards an increasing antibody-dependent enhancement risk because is focusing, skewing the response towards cross-reactive enhancing antibody like the one targeting the fusion loop. This was TB vaccination, but fortunately, luckily, it's not true for all the vaccine. Different vaccine show different cross-priming. And here I'm showing you the results of cross-reactivity after TB vaccination and after yellow fever vaccination. So if we look our IgG fraction in our cohort baseline, we have the cohort here in pink that have been previously vaccinated with TB, which is able to bind and recognize all the flavivirus that we have tested, where the naive subject here in light blue do not react. After yellow fever vaccination, this cohort increased the reactivity, and the naive cohort has only a minimal cross-reactivity, and the response that is induced is focused against yellow fever. So if we compare this sera here and this sera here are basically sera coming after either TB vaccination or yellow fever vaccination. As you can appreciate, the cross-reactivity is different. With yellow fever, IgG generated after TB vaccination are mostly cross-reactive with several flaviviruses. And IgG generated after 17D vaccination are focused towards a specific response towards yellow fever. So to conclude what I presented you, pre-existing flavivirus immunity shapes flavivirus infection, not only in dengue, but for all the flaviviruses. So it means that this needs to be taken in account for vaccine design and also for immunization campaign, especially in endemic area where there is population differently primed by different infection with flavivirus. To do that, we need to know what is the immune status of this population, and we need to do the construction study to analyze the complexity of the human polyclonal response to flavivirus, and have a more global approach to flavivirus vaccination, design a campaign with preferential sequential immunization, vaccine design that does not present epitope that are inducing enhancing antibody, not PRM and fusion loop exposure. The basal immune status is important, cross-reactive presence of cross-reactive antibody. Also the T-cell immunity, have a vaccine that maybe stimulate more T-cell immunity. T-cell immunity has been shown also in yellow fever. We have shown that high level of T follicular helper cell or activated CD4 are positively correlated with a good antibody response. T-cell response, importance of T-cell response in other flavivirus has been demonstrated. So there is all the interest to have a vaccine that stimulate also the T-cell immunity. Going back to what I told you of the primary response between vaccine and the difference between TB and yellow fever. So we noticed the yellow fever is a particular optimal response, they skewed the response to a protective immunity. And so we wanted to study this more in depth, and this is the subject of our EU project, the Yellow for Flavi, that was to investigate the role of the viral structure of the viral particle, the structure of 17D, because we think that has a role in presenting preferentially protective epitope. Our consortium involves 13 partners with country in Europe, and we have also a partner in Colombia. And we are going to study from one side the virus, so the structure of the viral particle, the role that this structure may have in intracellular trafficking, antigen processing, and antigen presentation. We have all collaborator immunologists that will study the host response to the vaccine, starting from the site of injection of the vaccine, from the skin at the level, what happened at the level of the skin, to the development of B-cell immunity and T-cell immunity to establish long-term memory. And in the consortium also, we wanted to look at vaccine in a more extended way. At the population level, we have a group of social scientists that will study the communication strategy and approach the vaccine hesitancy and the impact that this has on the population acceptance of vaccine. This is the consortium more in detail with the different partners from the different nations, and we have now a new entry that is Omniscope, which is a company that will work together with the other company that we had, ImmunoWatch, to predict and identify B-cell and T-cell epitope. In the consortium, they join us with the open call that had partner to an EU Horizon grant. I want to spend now the last few slides just to tell you a little bit more what we are doing in the consortium, in the work package, and how we want to study the antigen structure. We want to look at the way how the envelope protein, in particular 17D, is presenting epitope at the surface of the viral particle, and the molecular determinant that establish this structure are derived for the interaction not only of the E protein, but the interaction with the premembrane protein. So to study the interaction between the two proteins, we do this in the context of the viral particle and not of the isolated protein by X-ray crystallography, and we use cryo-EM. So we have developed a protocol to study all viral particles by cryo-EM, which at the beginning involved the inactivation of the virus. So this is because our Titan microscope is in a BSL-1 facility, and Flavivirus are BSL-3, and even the vaccine 17D is BSL-2. So we have developed a protocol to inactivate the virus in a way that we do not affect the conformation of the structure of the virus. And we have now two different types of protocols that we use to study the maturation of the virus in the cell in situ. We can use chemical inactivation, showing you here West Nile virus in infected cells. We can detected by cryo-electron tomography. But to study the epitope exposure and the interaction with antibody, we look at the purified virus, and we use for that UV inactivation by irradiation. We can inactivate the virus, but we can test by functional study that we have not impacted the conformational structure of the envelope protein, because if we take the purified virus and we mix with fluorescent liposome and we lower the pH, we can induce conformational change, fusion, lipid mixing, and by mixing the membrane of the virus with the fluorescent liposome, the dye that is in the liposome is dequenched, and we have a signal. So we can, with this functional in vitro fusion assay, detect viral fusion. And if we use in this assay UV inactivated virus, we are still able to detect fusion, which is telling us that we have inactivated the virus, yes, but we do not have impacted the conformational changes that occur at the envelope protein. And so we can study interaction with antibody. This is just an image that we can have for the virus with in complex with a neutralizing antibody, where we can map the antibody on the virus. We can identify the epitope where the antibody binds, but more importantly, we can study the stoichiometry of the virus, of the antibody binding, and how the antibody is mediated inactivation or neutralization of the virus. And I think I'll stop here. I just want to thank the funding that support our research. This is my group with a new arrival, Nilanchu, Stefan, and Julien, where Max was a postdoc that has worked also on other viruses in the group, now left and is at Sanofi. This is the rest of the group, and Marino Urarde is the project manager for the EU project. And I thank you for your attention.
Thank you very much. Giovanna, now we have time for questions. So if you can address some question, please use the chat.
Maybe I stop sharing, and if there is need, I will.
Okay, thank you.
So that we see each other. I don't know.
I have a question for you. Regarding to your result about the increase in the AGC that can mediate AD after TBE vaccination, but you are not observing that in the opposite when you are using sample from yellow fever vaccinated individual. My question is, do you think that it's possible to know in advance which kind of flavivirus can generate this behavior after immunization? This is something particular for this TBE virus, or we can anticipate that other flavivirus can have a similar behavior?
Other flavivirus, other vaccine can have this the same behavior of TBE. For example, it has been reported for Japanese encephalitis vaccine, GEV. There was a study where reported GEV vaccination was also basically doing the same thing that what we're seeing here. Sera of GEV vaccine were showing ADE in vitro for dengue. Now, we don't know exactly what is causing that and why 17D is different compared. It is linked to the structure of the particle, probably of the epitope that are presented. The one difference, important difference between the two vaccines is the fact that TB is an inactivated vaccine, where 17D is a live attenuated virus. And it's possible that the TB inactivated vaccine is more heterogeneous and it's presenting preferentially fusion loop. Epitope is, the particle is more open. And the GV in the study that reported the ADE was also an inactivated. For GV, there are two different types of vaccine, one live attenuated, one inactivated, and the one of the study where they reported ADE was inactivated. So it could be, it is basically the heterogeneity of the particle. So maybe studying the heterogeneity of the vaccine can predict the type of response that we may induce.
Okay, thank you. Someone has any question? If not, I can continue because I found this presentation very interesting. So my second question is regarding to the AD. What is the impact of this ADE immunity that you are observing in the lab in the real life? I mean, because as you know, the final outcome would depend on the balance between the neutralizing antibody that we are generating and also this antibody that can mediate ADE.
Yeah.
Also, we have another actor in the field, that is the T cell response. So we know that the T cell response can be very helpful avoiding the development of severe disease. So
absolutely,
how we can integrate all this part in the final picture?
So concerning the ADE, we had to do experiment in vitro because in our cohort, we couldn't find subjects that got the TB vaccination, then the yellow fever vaccination, then they went to, they go to South America and get dengue. It was very difficult. So we had to do the study in vitro, but I mean, ADE is the increase of exacerbation of disease has been taken into account for the dengue vaccine. Dengvaxia, for example, has been now discontinued because it has been seen that in naive population, the subject, the children were vaccinated with Dengvaxia, then had a higher risk of hospitalization after a vaccination. And so this was not what you want from a vaccine. And this is why now the vaccine is given, was suggested to be given to people who already had pre-immunity and from this type of vaccination worldwide and the number that you have to vaccinate, the fact that you need to screen all the subjects before to give the vaccine is not what you want, basically. In terms of T-cell immunity, this is known. We, in our experience, we have seen in our study of a cohort of 17D vaccination that T follicular helping circulation are positively correlated with the, for example, and also in a recent paper that published by the group of Simon Rothenfusser, also the CD4 activation as important to have a strong antibody response. But also T-cell immunity in general is important because there was also recent work for a group in Singapore where they showed that if you use a chimera 17D, where you switch you put other envelope protein for other flavivirus, you may still have protection even if you remove the antibody, the envelope protein that is the target from the neutralizing antibody, if you have a good T-cell immunity. So definitely in a design for a vaccine, we need to consider the epitope, definitely, that we are going to put. So we don't want to put epitope that induce ADE, but also we need to have elements that will stimulate positively the T-cell immunity.
Okay, thank you.
There is a question? Mm-hmm. I saw just the hand, but I didn't see the question because I have to open the chat, maybe would be more useful.
Could you please address the question by the chat?
So you also be able to see screenshot?
Cannot see
No,
any.
I don't see it. I don't see it in the chat.
No, I cannot see any question in the chat.
Because my understanding was that the public is muted, so you can raise your hand, but you need to write the question in the chat.
Yeah, exactly. Yeah, you pay attention on the antibody that are targeting the fusion peptide. Do you think that there is other region in the envelope glycoprotein that can also mediate this kind of ADE antibodies in addition to the fusion peptide?
Well, not only the fusion peptide, there's also this PRM antibody are known also to induce ADE, and then there it depends again from the structure of your particle. If a virus is more mature, you will have less exposure of this epitope, but if you have a virus that have more they are less efficient in the maturation, you will have more heterogeneity, and so you will have other epitopes that are expressed that are targeted by this ADE-inducing antibody.
Okay, thank you.
Okay, now there is apparently in the chat, you can ask your question by the microphone.
Yeah, directly by the microphone.
Okay, then I'll take advantage of that. I'm Federico Bertoglio from Technische Universität Braunschweig in Germany. Thank you very much for the very, very interesting talk. It's more a curiosity that I have. So since already the natural virus produces half in nature particles, do you think it's even possible to get for example, a virus-like particle vaccine which has completely nature E protein to avoid to get these ADE antibodies?
Yeah, I think this is the way towards where we want to go in designing a vaccine, where we want to maybe eliminate the presence of PRM in the vaccine, and so have maybe only ME, so you don't have this immature. There is a technical difficulty in that because you will not have production of particles, but you may think as an mRNA vaccine, for example, that will express this epitope and then you will avoid generation of antibody against PR in that case. But this is something that is at the study at the moment is really preliminary. Or have a way to produce virus-like particle that have a particular structure where you enhance the maturation process, and so you don't have PRM, and you have only mature particle. This is another approach.
Thank you so much. Oh, I guess I could also appear on the camera. Yes. Okay. Now, yeah. Thank you so much. Yes. Very, very interesting to think to try to with not having PRM in the vaccines or in the VLPs, but it's tricky.
Yeah, it's tricky.
Yeah, we have a question in the chat from Alejandro Vargas, and the question say, could cross-reactivity be expected with other arboviruses? More broadly, how could this evidence impact vaccination schedule for other viruses?
Other arbovirus, not Flavi, Alejandro? I mean, I think cross-reactivity, it has not been described, cross-reactivity outside the Flavi in that. I think there are already so many Flavivirus co-circulating in endemic area that is already a big problem for that. Then if this may impact chikungunya circulation, for example, it's an open question. It's definitely, every infection shape the immune response, and so it may leave some trace.
More question? I have a question regarding to the ADE activity that you observe in the IgG fraction of these antibodies. You know the IgE can also be mediated by complement through complement receptor on the surface of susceptible cell. Do you think that in this how is the impact of both the FC receptor and also the complement receptor in this final outcome? Do you think that we need to pay attention also to complement when we are analyzing this ADC activity of the IgG in plasma or IgN maybe in plasma?
Well, usually it has been seen the effector function of the FC of the antibody has been seen as positively because you can mediate a different type of response that can result in destruction of the infected cells, so it's more positive. Here, the entry via the FC receptor, because it's like mimicking an entry in a cell where there is no receptor for the virus, but it enters via the FC receptor, and the fact that the complex is not totally neutralized, it brings really, it's like a Trojan horse and brings the virus in the cell, where maybe complement may be more efficient in destroying in that case.
Okay, we have another question in the chat. That is, can cross-reactivity be reduced or knockout?
You mean knockout the sequence of fusion fusion loop, probably? The problem that we have is, for example, if we want to design a vaccine where you don't want to induce fusion loop antibody, but you want to induce ED antibody, E-dimer-dependent antibody. Now, the fusion loop is part of the epitope of the ED too. So, so in effect, our depletion, for example, when we have this locked dimer, where we put the mutation on the ED epitope, it's true the dimer cannot open, does not expose the fusion loop, but at the same time, the mutation that we had to introduce to generate the locked dimer affected one amino acid that is recognized by some of the ED antibody. So this construct was not able to deplete all the ED antibodies. Some they are still able to bind, but some other are not. And it's telling us that if we manipulate the fusion loop, we are affecting not only the fusion loop antibody, we affect also other antibody that in reality we want to keep.
Okay, thank you. So if we don't have more questions, I would like to share with you briefly. Let me see. Let me see if I can. Can you see a slide in the?
Yes.
Yeah. Okay. This slide just summarizing the next talk that we are going to have in this seminar series. The next one is 10 of April by Julien Pompon from Montpellier. And then we can move to May, where Professor Luisa Barson will give a talk. We don't know exactly what is the title of the talk. And then in June, Mireia Pellegrin and Mar Naranjo will share with us result and new thought about the use of antibody as new to understand the use of this antibody to enhance the immune response against different antigens. And on September, we have fixed the talk by Miguel Ángel Martín Aceves about the metabolic reprogramming during West Nile virus infection. If no more question if we don't have more question, I would like to finish here this seminar. Again, thank you very much, Giovanna, for your talk and your participation in this seminar series, and thank you to all of you for this time and to share this amazing presentation. Thank you very much.
Thank you, everyone. Bye.
Bye-bye.
Bye-bye. Bye.